Hello, and welcome to the tour of photosynthesis. Today, you will learn the ins and outs of photosynthesis from our very own workers. But first, I would like to give you a rundown of the facility, also known as the chloroplast. The chloroplast is the site of photosynthesis of all plants. It is mainly found in mesophyll cells, which make up the tissue in the interior of a leaf. One part of the chloroplast is the stomata. These act like microscopic windows of our facility and allow carbon dioxide to enter the chloroplast and oxygen to leave. Additionally, the chloroplast has an envelope of two membranes that surround a dense fluid called stroma. Then within the stroma, there are tiny sacs called thylakoids, which separate the stroma and the thylakoid space. Also, when thylakoids are stacked into columns, we call them grana. Finally, we conclude our brief tour with the color of the chloroplast. This green pigment is called chlorophyll, and it is found in the thylakoid membranes of the chloroplast. Before we talk to the workers here, we should go over the overall formula for photosynthesis. The general equation is 6 carbon dioxide molecules plus 6 water molecules plus light energy creates 1 molecule of glucose and 6 molecules of oxygen. It is important to know that the oxygen released in this process is produced from the electrons of the water and not the carbon dioxide. Additionally, photosynthesis is an endergonic reaction, which means that it requires energy. Electrons increase in potential energy as they move from water to sugar. Therefore, this process needs energy, which it gets from light. Next, we will talk with an electron worker about their role in photosynthesis.
As an electron, I am heavily involved in photosynthesis. Before I start on the process of what I go through, called light reactions, I'm going to tell you a little bit about light. Light is an electromagnetic wave that can be seen by the human eye. Visible light exists from wavelengths, the distance between crests of electromagnetic waves, of about 380 to 750 nanometers on the electromagnetic spectrum, which is the entire range of radiation. Photons, the particles within light, have fixed amount of energy that are inversely related to wavelength. The shorter wavelengths have photons with greater amounts of energy. A pigment is a substance that can absorb visible light. Different pigments absorb light of different wavelengths, and the chlorophyll is the main pigment found inside chloroplast. In photosynthesis, the chlorophyll of chloroplast absorbs the energy of light, and I am able to become excited and jump to a higher orbital. However, my excited state is very unstable, and I will quickly fall back to my ground state. As I fall, another lucky electron in a nearby pigment molecule is raised to an excited state. This process continues until the energy reaches the P680 part of the special pair of chlorophyll A molecules within the photosystem II reaction center complex. The energy excites an electron, me, to a higher energy state. I am then transferred from the excited P680 to the primary electron acceptor. An enzyme then catalyzes the splitting of water into its components, and the electron replaces me as I leave photosystem II and make my way to photosystem I. I travel on an electron transport chain and fall to lower energy states, providing energy for the pumping of protons. Chemiosmosis then produces ATP through the creation of this proton gradient. While I traveled, light energy was transferred via harvesting complex pigments to the PS1 reaction center. Electrons, like myself, are passed in a series of redox reactions from the primary electron acceptor of PS1 down a second electron transport chain through the proton ferredoxin. We are then transferred to NADP+ to create NADPH via the enzyme NADP reductase. That's what I like to call linear electron flow. There's also cyclic electron flow, in which I cycle back from the ferredoxin to the cytochrome complex, and from there go right to the P700 chlorophyll A in the PS1 reaction center complex, never seeing photosystem II.
Now that we've produced ATP and NADPH during the light reactions, we're going to proceed to the Calvin cycle, where this energy from light is going to be converted to chemical energy in a sugar molecule. So we're going to start with a CO2 molecule from the atmosphere. Now this CO2 molecule, or this beginning part of the reaction, is going to occur 3 times over, but I'm just going to explain it one at a time. So you're going to start with one CO2 molecule, and it's going to come into the reaction and bind to a 5-carbon molecule called ribulose biphosphate, or RuBP. So this CO2 with the RuBP is going to form 6-carbon molecule that has a phosphate on each side. This molecule is really unstable, so it's going to immediately split into 2 3-carbon molecules with a phosphate on each end. This molecule is called 3-phosphoglycerate. Now, with these 2 molecules of 3-phosphoglycerate, we're going to add another phosphate group to each side so that it's a 1,3 biphosphoglycerate. So with this NADPH from the light reactions, it's going to donate a pair of electrons, of high energy electrons, to each of these 3 carbon molecules because we have 2 molecules of NADPH. At the same time, 2 phosphate groups are going to be released from the molecule so that we now, because this is occurring 3 times over, we're now going to have 6 molecules of a 3 carbon molecule with a phosphate group on each end, and the phosphate group's attached to the 3rd carbon. So we have 6 of these molecules. This is called glyceraldehyde 3-phosphate, or G3P. And then this 3 times over reaction is going to happen twice because this G3P molecule is going to be released. And once you have 2 of this 3 carbon molecules, you can produce the 6 carbon molecule of glucose with a phosphate group on each end. Now, because this is a cycle, we have to regenerate the RuBP that we started with. And to do that, now that we've donated 1 of the 3 carbon molecules, we're going to continue on and we're going to have 5 3 carbon molecules with a phosphate group on the end left. Then 3 phosphate groups are going to be added to the molecule, and then we're going to have 3 RuBPs, which is a 5 carbon molecule with a phosphate group on each end, which is what we started with.
As a carbon dioxide molecule, I enter through the stomata of leaves for photosynthesis, but in hot, dry environments, those leaves close up their stomata to keep in water. It can be hard for me to enter, and then oxygen produced by the Calvin cycle takes my place with rubisco. This wastes energy to make more carbon dioxide instead of helping to create ATP and sugar, like I do. This is called photorespiration. So how is the oxygen kept out? In plants called C4 plants, I join a molecule called PEP in the mesophyll cells. Together, we have a higher affinity for carbon dioxide for the Calvin cycle and no affinity for oxygen. We then move to the bundle sheath cells, and I am released for integration into the Calvin cycle. No oxygen used. Regular photosynthesis takes place only in mesophyll cells, but this change in location allows only CO2 to be pumped into the cycle area, so oxygen is not taken up by rubisco. In CAM plants, everything happens in the mesophyll cells, but I come through the stomata at night instead of during the day, when light intensity is high. I am then stored in organic acids in the plant's vacuole, and I am released into the Calvin cycle during the day.